Welcome back to Study Tips for IGCSE Biology. Today's is a continuation from the previous topic of inheritance. And without further delay, let's begin. In this chapter, you will study about sex linkage and DNA and protein synthesis. Sex determination. So in this slide is actually what is trying to explain about the diagram A and diagram B on the left-hand side illustrate the pair of chromosomes are responsible for determining what sex a person will be. They are called sex chromosomes. Refer to diagram C. A woman's chromosomes are both alike and are called X chromosome, and she has the genotype XX. A man only has one X chromosome. The other smaller one is a Y chromosome. So he has genotype XY. First, you have to write it down, the phenotype and genotypes of the parents. So you can see the parents' phenotype is a male and female, and the parents' genotype XY for male and female is XX. And gametes you have Alice X and Alice Y, and for female you have Alice X. And secondly, you have to identify the offspring genotype and phenotype, where when XX fertilized with the sperm's X, it will contribute female XX. And when XX fertilized with the Y, and it will contribute XY, which is male. So as a conclusion, we can say that we could expect ratio of female to male would be 1:1. Sex linkage. The X and Y chromosome determine sex and have genes on them. Most chromosomes have 2 copies of each one, a homologous pair. They contain genes in same position, meaning they have 2 copies of each gene. The Y chromosome is tiny and only have few genes. The X chromosome is much larger and have many genes, meaning for most of genes on X chromosome have only 1 copy. There is no second copy of Y chromosome. There are a few genes on Y chromosome that are not found on X chromosome, meaning that woman never has a copy of this gene and a man has only 1 copy. Genes that are found only on the non-homologous part of the X and Y chromosome are called sex-linked genes. One of these sex-linked genes control the productions of 3 different kinds of cone cells in the retina. A recessive allele of this gene B result in only 2 types of cone cells being made. And a person who is homozygous for this alleles cannot tell the differences between red and green. This is called red-green color blind. This situation is much more common in men than a woman. Sex-linked characteristic, a characteristic in which the gene responsible is located on a sex chromosome, which make it more common in one sex than in the other. Sex linkage. If you can see from this diagram, the green color blindness genotype and phenotype. Diagram above shows the various genotype and phenotype for this sex-linked condition. The green color is actually represent 3 possible genotypes that the woman may have, but the red color is actually represent 2 possible genotypes for a man. So, as what you have already learned, the questions to answer to this question is, what might happen if a woman who is a carrier for a red-green color blindness marry a man with normal vision? So first of all, you have to write it down, the phenotypes and genotypes of the parents, which is already done on the left-hand side. Genotypes for the LA capital B, LA capital B is a represent woman with normal vision. LA capital B, LA capital small b is a woman with normal vision who is a carrier. LA small b, LA small b is a woman with a red-green color blindness. You can see also from this diagram. And for the man with normal vision, this is a normal vision. So what it has is only LA B, capital B, and Y. And for the red-green colorblind male, is have LA small b with a Y. And secondly, you have to create the parents' phenotype, which is for normal man and carrier woman. Parents' genotype for normal man is LA capital BY. For the carrier woman, LA capital B, LA small b. For gametes, for the normal man, is have LA capital B or Y. And for the carrier woman is LA capital B and LA small b. And then thirdly, you have to identify the offspring genotype and phenotypes. Eggs that you already know that it have LA capital B and LA small b, whereas sperm consists of LA capital B and Y. So output from this table, you can make the conclusions that about half of their male children will be red-green colorblind, and all female children will have normal vision. DNA and protein synthesis. Chromosomes are made of DNA. DNA molecule is made up of 2 long strands of molecule called nucleotides. There are 4 different nucleotides, A, C, T, and G. Protein molecules are made up of long-chain amino acids. There are 20 different amino acids, and sequence of amino acids in protein molecule determine the final shapes of molecule. And the shapes of the molecule is actually affect how protein works. DNA contains a code which is determine exactly what sequence amino acids a cell should string together when it makes particular protein. This is how genes affect organism features. Protein affect features of organism. Many protein acting as enzyme. Enzyme determine the metabolic reactions. Other protein functions is as antibodies and receptor for neurotransmitters. A gene is a length of DNA coding for particular protein, and also genes determine what protein to be made. The genetic code. DNA has only 4 bases, A, C, T, and G. Protein have 20 different amino acids, meaning that 4 DNA letters have to combine to make different words each one signifying a particular amino acid. DNA bases are read in set of 3 called triplets. Example, the sequence CCG in DNA molecule stand for amino acid glycine, CAG stand for valine. So CCG, CAG tells the cell join valine amino acid to glycine amino acid. DNA is found in nucleus, as you can see from this diagram, where it is DNA located. And protein synthesis happen on ribosomes in cytoplasm. A messenger molecule called messenger mRNA is used to carry information from DNA to ribosome. Okay. When protein is to be made, mRNA molecule is made in nucleus, copying base sequence from appropriate length of DNA. So the mRNA move out from nucleus into cytoplasm and attach to ribosome. So as a result, if you have been eating a good diet, then the cytoplasm in your cell will contain plenty of all 20 different amino acids. As long as thin mRNA molecule passes through, ribosome link amino acids together in exactly the right order to make the desired protein following the code contained on mRNA molecule. With that, I would like to end my presentation. Thank you very much. I hope you enjoy studying, and please don't forget to subscribe to this channel. Thank you very much.